Big, exciting weekend? As exciting as it gets, I suppose. Let's finish up what we didn't finish up last time, which was the classification of enzymes. So biochemists are a little different than chemists. Chemists have a periodic table that they can point to all the elements and organize everything very nicely. And so biochemists have always envied that sort of thing with chemists. And one of the attempts to systematically deal with some of the issues or concepts in biochemistry was to systematically organize the enzymes. And so there was an enzyme commission that sat down and said, well, if we think about all the different kinds of enzymes and the reactions that they categorize, how can we classify them and how can we organize them in some systematic fashion? So they came up with the EC system called the EC classification, which breaks all enzymes into 6 categories. And I'm going to go through those categories with you briefly to explain how those enzymes fit. Each enzyme correspondingly has a number that corresponds to it. So an enzyme, for example, that's an oxidoreductase will be in category 1. It might be in a subcategory that has to do with sugar metabolism or something, in which case it would be 1.1. And that sugar metabolism might be broken into, let's say, glucose metabolism, which might be 1.1.1, et cetera. So that an enzyme is systematically described by the EC system by a series of digits that ultimately, at the very top level, are the 6 categories that you see here. So we're not going to go through, obviously, every subcategory and so forth, because that would be kind of a waste of time. But I think you should know something about the categories of individual enzymes and be able to recognize, in general, if shown a reaction, what category any individual enzyme would fit into. So the first of these are the oxidoreductases, and they're probably the easiest category to understand because these enzymes all involve oxidation reduction, and consequently they all involve an electron carrier, NAD/NADH or FAD/FADH2. So very easy to recognize these. This is clearly an oxidation reduction reaction is occurring. On the left side, we see malate, which is an alpha alcohol, and on the right we see oxaloacetate, which is an alpha keto acid. Okay, so this reaction is catalyzed by malate dehydrogenase, and we'll talk about that when we talk about the citric acid cycle next term. So there's the key elements there shown, the NAD+ and the NADH. Another fairly easy to understand, in fact, most of the categories are pretty easy to understand, I think, looking at them. This is another easy to understand category, and that is of a transferase. So transferases transfer a functional group from one molecule to another, all right? And commonly that transfer involves a methyl or a phosphate. So in this reaction, we can see the catalytic action of hexokinase, which catalyzes the first reaction of glycolysis. And as I think I've explained in class before, in this reaction, a phosphate from ATP is transferred onto position 6 of glucose to make glucose 6-phosphate and then yield ADP. So we can clearly see the transfer from one molecule to another occurring here. The third enzyme involves are what are called hydrolyses. And these are enzymes that catalyze the hydrolysis of bonds. Hydrolysis, of course, referring to water. So these guys use water to break bonds. And the enzyme that I'll talk about later in the class today, chymotrypsin, is a hydrolase because it uses water to break peptide bonds. So all proteases fit into this category because it requires water to break the peptide bond. Other things besides proteases also are categorized as hydrolases. So schematically, we can see the reaction occurring here. We see the break of the bond occurring as a result of the action of water. So the peptide bond there is being broken. And we're left after that with a new alpha carboxyl and a new alpha amino group that are shown on the right. The fourth category is probably the hardest of the individual ones to understand. These are of what are called Lyases. And as it says, these enzymes catalyze the non-hydrolytic, non-oxidative breaking of bonds. Okay? Well, if you want to, in simple terms, think about it just simply splitting a molecule apart without oxidation, you would be on the right track. So this is another reaction that we'll talk about when we talk about the glyoxylate cycle, which is related to the citric acid cycle. And in this reaction, isocitrate, which is a 6-carbon molecule, is being broken into a 2-carbon and a 4-carbon molecule, glyoxylate and succinate. There's no oxidation that's occurring in this process. You don't see any NAD or any NADH. And we're simply splitting this molecule into 2 pieces. So if you think about it in those terms, it's fairly straightforward. A is going to B plus C. Okay? And the isomerases. So isomerases we'll talk about also later in the term. And isomerases are enzymes that catalyze the change. They rearrange molecules without oxidizing or doing anything else to them. They're simply rearranging them. So a very good example, again, comes from glycolysis, phosphoglucoisomerase. And phosphoglucoisomerase converts glucose 6-phosphate into fructose 6-phosphate. If you count up all the atoms, you'll see all the atoms are there. They don't disappear. The molecule just simply gets rearranged from one sugar into another sugar, and this is an isomerization. If we had an enzyme catalyzing, for example, a cis to a trans bond, which we have in some of the fatty acid reactions, those also would be categorized as isomerases. And last, this one's pretty straightforward to understand. Ligases are enzymes that join 2 molecules by making covalent bonds. Okay? So the example I pulled out here is from the urea cycle. In this reaction, citrulline and aspartate are being joined together using energy from ATP to make this molecule called argininosuccinate. Yep, there we go. And you see in this reaction, it's a little bit complicated, but on the left we have a molecule in green, which is aspartate, which is linked to, I'm sorry, I take it back. We've got ATP, sorry, which attaches to the citrulline. That adenylation of citrulline is an important step in the catalytic process. And then we see aspartate being added in after that intermediate has been formed. So aspartate is in the upper part in this sort of purplish color. And aspartate attacks that bond where the citrulline is attached to the adenosine monophosphate, releases adenosine monophosphate, and creates argininosuccinate. The overall net of this reaction is that citrulline and aspartic acid got joined together to make this molecule called argininosuccinate. You can see also, again, no oxidation occurring in this reaction. There's no NAD or FAD involvement. Okay, there they are, and there's the product. Okay, that was last time, so we're not going to sing a metabolic melody here. We have one later, relax. We will have one. Always have one. Okay, so the next thing I want to, next topic I want to go into, and I'll remind you that, of course, that material for the exam ended with the last of what I said on Friday. So everything I say today is not for the first exam, it's for the second exam. Okay? So that includes this, that includes what I just talked about just a minute ago. So enzyme catalytic mechanisms. And we spend a lot of time in this class talking about enzymes, and with good reason, because without enzymes, we don't exist. Enzymes speed reactions. Enzymes make it possible for living systems to exist as such. If we wait for some of the reactions to occur by a catalytic process independent of enzymes, we discover that, in fact, the reactions are just simply not occurring fast enough to sustain what we need as energy for life. So spending some time talking about catalytic mechanisms, how it is that enzymes catalyze reactions, is important. And I will say up front that I'm not much of a mechanism person. Okay? I just don't get much into mechanism, but there are mechanisms that we need to understand in order to understand function of enzymes and the catalysis that they're performing. So necessarily, I make the mechanisms that we go through relatively straightforward, relatively simple to understand, I hope, so as not to burden you with unnecessary mechanisms that really aren't essential for you. The prime mechanisms I'll be talking about today, at least, are those that involve proteases. Proteases are enzymes, as I said, that break peptide bonds. And proteases can be broken into various categories based on the mechanism that they use. So the biggest category that I'll be talking about are those of the serine proteases. And I'll spend a fair amount of time talking about how they work. Now, what I want you to do in seeing these mechanisms is to understand something about what's going on, but look at big picture in these, okay? I'm not asking you to do any significant pushing of electrons, for example, in order to understand these mechanisms, but there are important steps in the catalytic process that are important for you to understand, and I try to summarize those both in the slides and also in what I have to say. Well, the first enzyme I'm going to talk about is chymotrypsin. And chymotrypsin is the sort of poster child for the serine proteases. Now, serine proteases are enzymes that get their name based on the fact that they have within them a serine residue that is essential for catalysis. They contain a serine residue that is essential for catalysis. And as we will see, that serine residue plays an essential role in the catalytic process. Okay? So very important function. Now, proteases, as I said, cleave peptide bonds. Okay? They cleave peptide bonds. And so proteases are important for the process of digestion. Proteases are important as laboratory tools because sometimes we want to break a protein into smaller pieces so that we can work with the individual pieces more easily than we can with the entire protein. For example, if we're trying to determine the sequence or the identity of a given protein. And so those are very important features. We will see that proteases cleave peptide bonds generally with some specificity. Okay? Specificity, meaning that they will recognize specific amino acids within a protein sequence and cut adjacent to them. So chymotrypsin, as we will see, cuts adjacent to amino acids that are large and nonpolar, like phenylalanine, like tryptophan, like tyrosine. Okay? So there's specificity of cutting, as I said, and that specificity of cutting is due to the fact that the proteases, as we shall see, have specialized sites within them that will only bind the residues that I described. So, for example, chymotrypsin has a section of it that will only bind phenylalanine, tryptophan, or tyrosine. And that's a little misleading because there's some wobble, but basically that's what it prefers to bind at its binding site, or those three. So once it's bound the amino acid residue of a polypeptide chain at its binding site, then it simply cuts immediately adjacent to that, and we'll see how that occurs. So the serine proteases all have a common active site. They all have a common composition and structure. If I were to, for example, take the three-dimensional structure of trypsin and overlay it on top of chymotrypsin, you would see the two are almost, not quite, but almost identical in three-dimensional structure. Okay? Very, very little difference. And of course, that reinforces the idea that we have that structure is necessary for function, and structure implies function. Trypsin, just like chymotrypsin, is a serine protease, and it uses a mechanism almost identical, or actually it is identical to that of chymotrypsin. Okay. And the last reason for bringing these up is that these enzymes are mechanistically very well studied. Okay? So we study them because they're well studied. I guess that's kind of a silly thing to say, but these enzymes are very good examples for you to learn mechanism because of the fact that we have a lot of knowledge about them. Okay, so chymotrypsin, as I said, is a protease. And chymotrypsin, if we had a polypeptide chain that looked like this, glycine, phenylalanine, aspartic acid, threonine, alanine, asparagine, what we would see is that in the presence of the enzyme and in the presence of water, the bond on the carboxyl side of phenylalanine is cleaved. So I said phenylalanine, tryptophan, tyrosine. Okay? The result of that is we started with one polypeptide chain on the top, and we now have two polypeptide chains on the bottom. Okay? So that bond has been broken. And it's important to note that it's on the carboxyl side, meaning that when the bond is broken, the phenylalanine is there, and its carboxyl group remains intact. If we had cut on the amino side, we would have cut on the nitrogen side of the molecule. Okay? Some proteases cut on the amino side, some proteases cut on the carboxyl side. So recognizing that difference in terms of specificity is important. Now, chymotrypsin, as I said, is a little bit of a bad example because chymotrypsin is an enzyme that's fairly flexible in the substrates that it will bind to. It will also, for example, accept threonine to a limited extent. So it's not a rigid, specific, it's only going to bind one thing and act on it, but it will strongly prefer the larger nonpolar amino acid side chains like the three that I mentioned. And for our purposes, we'll just say that those three are the three that it prefers to bind to. Okay, well, I noted that chymotrypsin is flexible in terms of what it binds to, and that flexibility turns out to be quite advantageous for us to be able to study how it is that chymotrypsin actually works. So I like to say that biochemists are lazy people, right? I'm a biochemist, so I can say that about myself. Biochemists are lazy people, and we like to find easy ways to do things. And so if we want to study mechanism, it's really nice if we have a molecule that gives us some kind of an indication when it's been cut. The polypeptide chain I showed in the last one doesn't give me any indication that it's been cut until I analyze it on a gel. And that takes a long time for me to do. So if I have a reagent that chymotrypsin will recognize, bind to, and cut, and when it cuts it, it creates a color, then I can instantaneously tell if the cutting has occurred. So what you see on the screen is a molecule that fits that criterion. So if we look at it at first glance, it looks kind of like, well, it's got like a phenylalanine ring sticking up above it, and it doesn't look like it's attached to a polypeptide chain. That green bond, for example, isn't one that we would see within a polypeptide chain. Okay? However, chymotrypsin is accommodating. Chymotrypsin says, well, it looks enough like a peptide to me, and it will cut it. And so when it cuts it, what it does, again, using water, you see the plus water on the left side there, you see that it splits it into two pieces. One piece on the left we don't even bother to name, and the piece on the right, paranitrophenolate. Now, paranitrophenolate has the property that it's yellow. It's a very bright yellow. So that means then I start the reaction. When I add my chymotrypsin to this material, I have no color whatsoever. And then as the reaction proceeds, I get more and more yellow. Well, color, of course, I can easily and quickly monitor in real time with a spectrophotometer. So doing that, I can then understand the steps or how this process is occurring. And when I do that, I find something kind of interesting, which is what I'm going to show you next. Okay? So everybody understands that the cutting produces a yellow color, correct? Anybody awake? I talked to the person who taught the last class, and she said the whole class was just like, ugh. And I don't want to say this about my class, but you guys don't look like you're the most energetic today. I will say that. You kind of look like it was a tough weekend. Studying biochemistry? Who would have thought, right? Okay. All right. So anyway, the point is that cutting this molecule into two pieces produces one molecule that has a color, and we monitor the production of that color. Now, If we do that, we see something interesting, and the something interesting that we see is here. All right? What we see is that this thing goes through a very rapid initial catalysis, and then it kind of levels off. Now notice that what we're doing here is not substrate concentration on the x-axis, but we're looking at time. We haven't had a plot like this so far. Okay, we're looking at time. On the y-axis, we're looking at the amount of color produced, and we can measure that using a spectrophotometer. Why do we have that bend that occurs up there? Well, when people first did this experiment and they saw that bend, they realized that the reaction must be occurring in two steps. Two steps. A fast phase and a slow phase. And that transition that you see there is moving from the fast phase to the slow phase, which gets things spread out over time. Okay? Now, when I go through and show you the mechanism, I'm going to show you where that fast phase occurs and where that slow phase occurs. But I'll tell you in advance what's actually happening. What happens is, in the fast phase, the peptide bond is broken. Okay, no surprise. Well, that's what the enzyme is supposed to do, right? The peptide bond gets broken. However, the product of that catalysis is that part of the polypeptide chain becomes covalently attached to the enzyme. So part of that catalysis, the polypeptide chain becomes attached to the enzyme. The other part of the polypeptide chain, the part that was cut, is released. So in the case of the example I showed you, the part that's released is the yellow part. It goes flying away. Okay? The slow part of the catalysis is the release of the covalently bonded polypeptide chain from the enzyme. And that takes a longer period of time to occur than the initial breaking and release. Okay? So those are the two steps that we're going to see. And I'm going to show you in some detail now how the mechanism of this reaction proceeds. Okay? All right. Well, yes, question? So, in the slow phase, the polypeptide chain has to be released from the chymotrypsin, and we'll see that the environment in which it exists within the enzyme is favorable when water enters. But we'll see that schematically. So I just want you to see big picture, the fact that it's getting physically attached. Okay? Other questions? Okay. So, another question. Yeah? Yes. Okay. Yeah, common question. So he says, why, after I see the release of the first one, does the color continue to go up? Or why does it even change? Okay. What's happening here is that the enzyme in that quick phase is releasing the yellow. Some that finish really quickly, like those down at the far bottom left, have enough time to release the second part and actually start the reaction again. So we're not seeing one reaction for any enzyme. We're seeing the sum of all the reactions of all of the enzymes. And they actually get spread out over time. And that's why we don't see a distinct boink like that, but that rather it curves up like that. So what we're seeing is a mix of enzymes that have catalyzed one reaction, two reactions, three reactions, et cetera. Does that make sense? Okay, so everybody's starting at the same point, though. Bang. And in that bang phase, that's why it goes up so quickly. Then they more slowly go into the second phase, and that mixes things up a bit. Make sense? Okay. Well, let's take a look at how this enzyme works. And we're going to look inside the active site of the enzyme, up close and personal. Okay? We start with the substrate. And the substrate is a polypeptide chain that you can see on the left. That polypeptide chain has the R there refers to a long string of amino acids. It's not just the side chain of one amino acid, but rather the whole string of amino acids. And the R prime on the other side is the other string of amino acids that also each have individual R groups. In this particular case, let's say phenylalanine is the R group of the first amino acid on the molecule on the left side. So the active site binds to that phenylalanine in the catalytic process. We can see that here. Now, looking inside the active site, we see 3 amino acids that are essential for the catalytic action of serine proteases. They're called the catalytic triad. Yes, that's an important term, the catalytic triad. The catalytic triad consists of the amino acids serine, histidine, and aspartic acid. Serine, histidine, and aspartic acid. Those amino acids are not close in primary sequence. How did they get to be close? What process? Folding. So the enzyme had to fold to bring these guys into close proximity and create the active site. Now the active site, you remember, is the place where the reaction occurs. It's adjacent to the binding site. Okay, we're going to see it's adjacent to another site, inside of the enzyme. All right? But the active site is the exact location where the reaction takes place. And we're going to see that serine plays a central role in the process. Okay. So there's the folded polypeptide chain of the enzyme. Okay. Can you tell I drew that figure? Probably can. Okay. There's the catalytic triad. Now, that catalytic triad is an important consideration for this enzyme. All right? Obviously, I said that's where the reaction is catalyzed. Well, how and why is this important? Well, this catalytic triad is just sitting there inside the enzyme all the time. And you might say, well, why does the enzyme have any specificity? Why doesn't it just cut whenever it binds to any polypeptide chain, any place in the polypeptide chain? And the answer is very simple. First of all, the binding site for the substrate, which in this case is the side chain of a phenylalanine, will not accommodate, for example, glutamic acid. It won't accommodate alanine. It won't accommodate lysine. Okay? But it will accommodate these guys. So only certain things will be bound by the binding site for the substrate. Now, what I hope I've planted in your heads is that the binding of a molecule to a polypeptide chain has a very, very slight effect. The shape of the enzyme changes very slightly, right? And that slight change that happens with the enzyme, in this case, affects the 3 amino acid side chains of the polypeptide chain. When the proper substrate binds, like phenylalanine, these 3 amino acids are brought into much closer proximity, okay, than they were before. Why does that matter? Okay? Well, let's imagine, let's imagine, if you will, that the aspartic acid on the right side, we can see it's negatively charged, it's full of electrons, it's brought closer to the histidine. The histidine has what we call a sink of electrons. A sink of electrons. You can imagine what a sink of electrons would look like. It would be a basin that would hold a lot of electrons. And that's what this basin-like structure in histidine is like. Do those electrons like being next to a bunch of negative charges on the aspartic acid? Nope. So what happens to the electrons that are in that sink? Well, they're going to move to the other side of the sink. And the other side of the sink is where a beautiful little proton is sitting from serine. And protons are positively charged. So normally serine's proton doesn't come off very readily at all, but because we've created an unusual circumstance here, those electrons have pushed over there, and the proton says, whoa, I see some electrons. And so the proton comes off of the serine. That creates a very reactive oxygen, because the electrons it leaves behind. It creates a very reactive oxygen. We call that molecule, okay, an alkoxide ion. So we can see that process right here happening that I just described. You see the pushing over of the aspartic acid. You can see, you don't see it, but you can trust me that there's a movement of electrons that's happened in the histidine ring, and the proton is just about to be pulled off of serine. Now, one thing I didn't say was, what do we call the place where the phenylalanine side chain binds? It has a name. We call it the S1 pocket. And we'll see later in the lecture that it actually is a pocket. And that pocket is set up very nicely to hold the side chain of a phenylalanine. Okay. So the binding stimulates that slight structural change. The slight structural change causes the things to happen that I just described to you. Okay? All right. Now, in the next step, we can see the proton has been taken away. We're left behind with the alkoxide ion, which is the oxygen without the proton. The alkoxide ion is very reactive, and so the alkoxide ion we see plays an essential role in this catalytic process. An essential role. There's our substrate sitting there waiting for the alkoxide ion to do its thing. The alkoxide ion makes a nucleophilic attack on the polypeptide chain at the place indicated. Okay? Why does it do that? All right? Well, the carbon that's there is relatively electron poor. The oxygen has pulled electrons away from it. A nucleophilic attack involves something that's electron rich, looking for a nucleus, and that carbon is a perfect nucleus for it to attack. We see that attack occurring right here, and the result of that attack is to break a peptide bond. The break of the peptide bond, however, occurs in two steps. Okay? The first step of the process involves the intermediate that you see on the screen. The oxygen has attached itself to the carbon. And we see another pocket in that enzyme that I've identified now called the oxyanion hole. The oxyanion hole doesn't just magically appear, it's there the whole time, right? But the oxyanion hole provides an environment for a very unstable intermediate to fall apart. That's what it's doing. It's allowing an unstable intermediate to fall apart. The unstable intermediate is that tetrahedral structure that you see there. That carbon in the middle has 4 bonds, and it's not a stable structure. If it's not contained in the oxyanion hole, that tetrahedral intermediate will covalently bind to the enzyme and not be able to be released. We want it to be able to bind only at the place that we want it to bind. And we'll see it's already done its first bond right here with the oxygen. So we don't want any more reacting of that carbon that's bound to the oxygen. So the oxyanion hole stabilizes that, and we'll see that this bond between the carbon and the nitrogen falls apart. So the oxyanion hole stabilizes the unstable intermediate and allows it to fall apart. Okay? All right. Now, the bond has fallen apart. It's broken apart. The first polypeptide chain is released. That was the half on the right. The half on the left, which contains the phenylalanine, is covalently attached to the serine residue. At this point, we have finished the fast phase of the reaction. Okay? Bang. That happens like just bang. It doesn't take any time to happen. It's happened. And in the case of the yellow intermediate that we had before, the yellow color would be produced at this point. Now we enter the slow phase of the catalytic process, and it's an essential phase because if we don't release that other polypeptide chain from the serine, the enzyme is dead. Right? Well, it turns out, as I said, the environment of the active site is such that water can come in, and we see water entering the active site here. And when water comes in, water looks an awful lot like to that histidine, like the serine's hydroxyl looked. All right? It looks an awful lot like that. So the histidine grabs one of water's protons and creates an activated hydroxyl intermediate. And that activated hydroxyl intermediate acts very much like that alkoxide ion that we had. It attacks, makes a nucleophilic attack on that same carbon, and the result of that action is going to release it. This step is slow. All right? Water's entered the active site. Nitrogen attacks the proton of water, pulls it off. The hydroxide attacks the carbonyl carbon. Okay? And we see the same tetrahedral intermediate that we saw before. But now when this one falls apart, it's going to fall apart at the serine because there's no other polypeptide chain attached to it. Okay? So we're stabling the intermediate. Oxygen is attracted to the proton that's on the histidine. Notice that's a positively charged proton there. The slow phase is complete at this point, and the second polypeptide is released. Now, that's a lot of information there, a lot of stuff there, a lot of steps there. I want to summarize what I've just said, all right? I want to summarize what I've said. Of course, the enzyme gets returned to its original state. All right, so what's happened? First, we had binding of the substrate to the S1 pocket. The binding of the substrate caused those changes I talked about. Those changes resulted in the formation of the alkoxide ion, and that alkoxide ion performed a nucleophilic attack on the carbonyl carbon of the polypeptide chain. The intermediate was stabilized in the process of the oxyanion hole. The result of that was the peptide bond was broken. It falls apart. The first part is done. The fast phase is complete. Next, water enters the active site, and that takes a while to get water into there. It's one of the reasons it's slow. And polypeptide 2 is released as a result of action of water, which is going through the same mechanism that was used in the alkoxide ion, and we're done. Now, look at that big picture. Look at it the way I've described the major points. As I said, I'm not pushing electrons, so you're not following electrons here at all. I want you to see what's happening in the process. Well, I said that the S1 pocket was the important binding site for chymotrypsin, and within the S1 pocket, we have the ability to bind large nonpolar amino acid side chains, and that I've tried to depict here. Okay, so the 3 amino acids that will fit into there nicely are phenylalanine, tryptophan, and tyrosine, and so that's why the chymotrypsin will cut adjacent to those 3 amino acids within a polypeptide chain. Other serine proteases, however, have different S1 pockets, and we can see a little bit here. Okay, here's one at a time. Here's the S1 pocket of trypsin. I told you trypsin was another serine protease, and trypsin is different. It has a structure very, very similar to chymotrypsin, but its S1 pocket's a little bit different. You can see that in the bottom of trypsin, there's a negatively charged aspartic acid. But what do negatively charged amino acids do? They favor positively charged amino acids. So the S1 pocket of trypsin favors the binding of lysine and arginine, and trypsin cuts adjacent to lysine and arginine. Here's the S1 pocket of another serine protease known as elastase. It has a narrow, nonpolar S1 pocket, and it likes to bind glycine, alanine, valine. Okay? So the nature of the S1 pocket is what's determining the specificity of the enzyme. The nature of the S1 pocket is what's determining the specificity of the enzyme. Now, how do we know about the catalytic triad? I've shown you a schematic figure, but how did we determine that that catalytic triad was essential and important? Well, people studying this enzyme decided to measure the importance of each of those 3 amino acids, serine, histidine, and aspartic acid. And they can do this by actually performing mutations that change one amino acid, the coding for one amino acid into another, and ultimately the protein that's made. So let me tell you show you what I'm talking about. Let's say if I take chymotrypsin and I take it no mutated form, I just take it as it is, and I measure its activity, and I give its activity a relative measure of 100. This is how active this enzyme is. I take the same enzyme and I make a mutation. If I change the serine to an alanine, how much activity do I get? Whoa. That's 7 orders of magnitude lower. All right? 7 orders of You might say, why do you have any activity at all? The answer is that, as you'll see in a little bit, if I had no enzyme at all, I still get a little bit of activity. Because, remember, an enzyme is not required to make all reactions occur. So this is 7 orders of magnitude lower. We know, therefore, that serine is essential for the action of this enzyme. If we convert histidine to alanine instead, what do we see? 7 orders of magnitude. That tells us that the histidine is essentially as important as the serine. And from what I've told you, that's no surprise, because the histidine is what's pulling the proton off. If I change the aspartic acid to an alanine, I see it's reduced, but not reduced quite as far as those other two. And it tells us that the role of aspartic acid isn't quite as important as that of histidine and serine. If I mutate all three, it's as bad as mutating only serine or only histidine, telling us that any one, either serine or histidine, I get rid of that, it's essentially fatal for the enzyme. And you say, what happens if you don't have any enzyme? There's the activity. Okay? I think it turned out to be 9 orders of magnitude. All right? But still a little bit of activity. All right, so that tells us then that the 3 amino acids that I described to you are essential for function. Reagents to cleave proteins are very, very useful in a laboratory environment. Okay, so I'm going through a few of these to show you, and then I'll next time talk a little bit about their mechanisms as well. Subtlysin cuts on the C-terminal side of large, uncharged amino acids, very much like chymotrypsin does. Chymotrypsin does the same thing. Trypsin, as I said, cuts on the C-terminal side of lysine and arginines, but not next to proline. If there's proline next to one of those, it won't do it. What would that tell you about what's happening at that proline? Come on, folks. What does proline do in a polypeptide chain? Creates a band. Maybe that band will prevent the binding properly, right? Okay? Think about the structure that we talked about. Structure and function, absolutely essential. Carboxypeptidase cuts on the N-terminal side of a C-terminal amino acid, meaning it starts at the C-terminus and sequentially chops off amino acids, one after the other, after the other, after the other. Okay? That's the one I'll show you here that cuts on the N-terminal side. Okay? And it's not specific in terms, actually it is a little bit specific. I'm simplifying the diagram here. But we can think of it as not being specific in the sense that all it cares about is that it cuts an amino acid off of the end. Elastase hydrolyzes the carboxyl side of small amino acids like glycine and alanine. And cyanogen bromide is a chemical that's used to cleave proteins. And that chemical hydrolyzes on the carboxyl side of methionine residues. And that's a very useful reagent because it's very specific for methionine. Okay, there's a polypeptide chain, and you can see some of the cleavage sites for the individual things that I've talked about there. Okay, well, I want to turn our attention briefly today, before I finish, to stopping the action of proteases. And this one is something that has a lot of medical relevance. There's a class of protein inhibitors known as serpins. And serpin stands for serine protease inhibitor. And serine protease inhibitors are polypeptides that basically block access to the active site by other polypeptides. So if I wanted to stop action of chymotrypsin, I would have a serpin that bound to it and prevented it from being able to bind any other polypeptides. If you stop binding, you completely turn off the enzyme. So that's what serpins do. They're polypeptide chains that bind and prevent an enzyme from working. In this case, serpins are for serine proteases. And they don't get cleaved themselves. You can imagine they have a structure such that they don't fit in the active site. Well, the one I want to talk to you about today is called alpha-1 antitrypsin. Alpha-1 antitrypsin is a very important, medically important serpin. Okay? It's a little misnamed. It was originally named because when the people who first studied it realized that it was inhibiting the protease known as trypsin. And it does inhibit trypsin. But more importantly, and more commonly, it inhibits a protease known as elastase. You've already seen elastase up there. Elastase is a protease in your lungs. It's common in your lungs. And having proteases in your lungs makes good sense because you'd sure like to be able to dissolve proteins in bacteria that you breathe in, right? As a way of controlling them. Having that there is important. However, elastase is made in fairly large quantities in your lungs, and if you have too much elastase, you get emphysema. So how does the body control that? Well, the body controls the amount of elastase by making alpha-1 antitrypsin. If it makes the proper amount of alpha-1 antitrypsin, it actually puts the elastase at the right level so that you're getting protection without getting the damage to the lungs that would occur by the elastase attacking your own tissue. Okay? Why is this thing medically important? Well, it's medically important because of what you see on the screen, reactive oxygen species. People who smoke generate a lot of reactive oxygen species. Okay? They generate a lot of things, but one of the worst things that they generate are reactive oxygen species. As their name suggests, they react very readily. When they react, they can produce a reactive oxygen species like what you see on the screen, a hydroxyl radical. And a hydroxyl radical will attack the sulfur of a methionine. And when it attacks it, it creates a sulfoxide, as you can see on the screen. Now, why do I tell you that? I tell you that because there's a critical methionine in alpha-1 antitrypsin that allows it to bind to elastase. And when that alpha-1 antitrypsin, when that methionine in alpha-1 antitrypsin gets oxidized in the lungs of a smoker, you can see what's going to happen here, right? It stops alpha-1 antitrypsin from being able to bind to elastase, and that's why smokers get emphysema, because the elastase attacks the lungs. Okay? There are people who have genetic disease or genetic defect or genetic disorder, whatever we want to call it, that don't make alpha-1 antitrypsin, or they're deficient in alpha-1 antitrypsin to some extent. And you can see a plot of the prevalence of deficiency of alpha-1 antitrypsin across Europe in this fashion. Okay? The worst there being, what is that? That's not Denmark. Is that Finland? In any event, okay? I should have looked at it before I put that up there. It was dumb of me, wasn't it? Okay. You don't want to have this because you're going to be much more likely to contract emphysema. Okay, well, I'm running out of time, so I'm not going to go through that. I will go through this. This, I hope, will reiterate everything I've said today. Enzyme of all proteases work almost identically using amino acids, triad catalytically. First they bind peptide substrates, holding on to them so tight, changing their structure when they get them in the S1 site. Then there are electron shifts at the active site. Serine gives up its proton as the reaction goes on. Next, the alkoxide ion, being so electron rich, grabs peptide's carbonyl group, breaks its bond without a hitch. So one piece is bound to it, the other gets set free. Water has to act next to let the final fragment loose. Then it's back where it started, waiting for a peptide chain that it can bind itself to. Go and start all over again. All right.